Today, I'm going to try to extract DNA from some tomatoes. But before that, you might be wondering why I was missing for the whole year. The thing is, it has been extremely busy for me for the whole year that even this video is actually a homework assignment. But anyway, let's continue. I first saw this experiment being done by Neil Red a few years ago, and I have always wanted to try recreating it. But at that time, it is not like everyone has alcohol at home, and now thanks to this pandemic, I now have the alcohol to do this. Let's first look at the anatomy of a plant cell. The DNA molecules are trapped inside the nucleus. So, to extract the DNA, I will need to break three barriers, namely the cell wall, cell membrane, and finally the nuclear membrane. To do that, I first have to prepare two things. First, I'm going to need some ice cold alcohol. To be honest, I don't even know what kind of alcohol this is, but what I know is this is definitely some kind of alcohol. I transferred some of it to a container and then put it into the refrigerator. Second, I'm going to need some soapy salt water, or if you like, salty salt water. I added some salt into a makeshift beaker and poured around half cup of water into it. Then I proceed to squeeze some dishwasher detergent and I just have to stir it. For now, it can be put aside. Next, I cut up some tomatoes into smaller pieces so that it will fit into the container. All I have to do now is to carefully smash them. I found that pen wasn't good enough at smashing things, so I switched to using a broken syringe. By doing this, I'm physically breaking the cell wall of the tomato cell, which is really the first step of getting into the cell. When I felt like it was good enough, I dumped in some of the soapy salt water into the container. At first, it might not seem like it is doing much thing, but the salt is actually dissolving the cell membrane of the cells. Remember how salt molecules look very similar to phospholipids. Some molecules have only one hydrophobic tail, but phospholipids has two. And this small structural difference causes it to form micelle structure in water instead of bilayer. However, they are still similar enough that some of the salt molecules will become the imposters and snatch the phospholipids from their bilayer structure, and thus ripping the cell membrane. Now, they just have to get into the nuclear membrane in the same way. Eventually, the DNA is released from the cells. You just don't see them now because they stay dissolved in water. The salt, on the other hand, provides sodium ions which will displace DNA protein from the DNA strand. After a few minutes of stirring and squishing, I proceed to filter the mixture to remove all the tomato junk. I used a piece of cloth as the filter and invert the bottle I cut as the filter funnel. Unfortunately, I don't have more transparent container so that you can see it passing through the filter, but this step is really boring and the filter keeps jamming that I need to wash it several times. Eventually, everything are filtered and I transfer the filtrate, which contains all the DNA we want, back into the washed transparent container. To further break down the proteins, I added some Jeff's spur, or juice from squished papaya leaves, which contain enzymes that will hydrolyze proteins. This further removes those DNA protein and free up the DNA. My teacher also told me that it contains enzyme for softening the cell wall. Now after all that hassles, all I have to do now is to carefully add some ice cold alcohol prepared earlier. Almost immediately, some stringy stuff is formed between the alcohol-water barrier. It is a bit crazy to think, but those things are actually DNA of the tomatoes. The reason this happens is because DNA is soluble in water but not in alcohol. so it precipitates out at a barrier where the solubility is the lowest. The cold temperature further decreased the solubility of DNA. I tried hooking the stringy DNA out using a bent copper wire because apparently I don't have a paperclip. But it is just not coming out as a strand. I was able to get a block of it sticking on the wire, and this is how it looks like. Just for fun, I decided to put it on my finger and see how that feels, but it instantly disappeared when I tried to rub it. Technically, I'm done with the experiment, so if you are my teacher watching this, you can now close it because I'm about to do things that are completely out of the procedures. Now that I'm done with the assignment, let's have some fun. First thing I want to try is to stir the mixture up. I have seen people dump the alcohol directly and stir it violently and it still precipitates out. However, in my case, it doesn't seem to be doing anything good. Some of the DNA just fell back into the solution and I'm getting nothing to stick on the copper wire. There might be several reasons I think this happened. First, I used an extremely small amount of tomato, like half of it, so there might not be much DNA to begin with. Besides, the temperature of the solution is no longer ice cold when I do this, and third, I might not be using enough alcohol. Anyway, since I still have a bunch of this solution lying around, I decided to dump them all in, or as much as the container can hold. I got this 3-minute time lapse which shows absolutely nothing but DNA moving around in the solution. I'm still not getting any stringy DNA out, but I was able to see a layer of DNA floating on top by shining light from behind. All I have to do now is to clean up all the mess. Wait, you think the video end? Not yet, one last thing. I tried putting the solution into the freezer and hopefully more DNA will precipitate out. I'm not entirely sure about this, but generally solubility decreases with temperature. And unsurprisingly, a bunch of things come out of the solution. To be honest, I'm not entirely sure if they are all DNA, because reducing the temperature would technically reduce solubility of everything. I look at one of the pieces under a macro lens, and this is how it looks like. Okay, I think that's it for this video. My teacher also gave us a 22 pages homework, so I have to do it after this. I'm probably going to go missing for another year, so see you soon.